Hey everyone, this is Dr. Mungli. Welcome to Dr. Mungli's channel. So in this video, I will be explaining you about degradation of sphingolipids. So before I take you into degradation of sphingolipids, I have a question for you. So the question is here. So the enzyme that is deficient in Tay Schs disease fails to remove which one of the following from GM2 gangliosides. So the choices, you have N-acetyl neuraminic acid, galactose, N-acetyl galactosamine, and sulfate. So I will give you answer towards the end of this video. Okay, let me take you into degradation of sphingolipids. So before that, let me quickly explain you what actually are sphingolipids. So sphingolipids, these are the basic structure of sphingolipid is ceramide. Ceramide is the basic structure of all sphingolipids. And this ceramide, as it is shown in the figure here, it is a combination of sphingosine and fatty acid. So the sphingosine and fatty acid combination, you're going to get ceramide. How do you get sphingosine? Sphingosine is synthesized from serine amino acid, hydroxyl containing amino acid serine. It is condensed with palmitoyl CoA by serine palmitoyl CoA transferase enzyme. That's how we make sphingosine in our cells. And this sphingosine with fatty acid, another fatty acid that is attached to a sphingosine, so we get ceramide. Now to the ceramide, a phosphoric acid which is added, and to that phosphoric acid, a choline is added. So basically, this is a structure of sphingomyelin. Sphingomyelin is a combination of ceramide and phosphocholine. So sphingomyelin is an example for sphingolipid. Okay. Some of the sphingolipids, they also contain N-acetyl neuraminic acid. Sphingolipids contain N-acetyl neuraminic acid. Let me just erase this. And whenever a sphingolipids, they contain N-acetyl neuraminic acid, so that type of sphingolipids, we call them as gangliosides. Okay. So example, we have ceramide with glucose, right? So that is glucosyl ceramide. To that glucose, galactose is added. So it is ceramide plus disaccharide. Usually we mention ceramide plus disaccharides as globosides. Ceramide plus monosaccharides are cerebrosides. Cerebrosides are ceramide plus a monosaccharide, glucose, galactose, like that. If it is a disaccharide, like what is shown in the figure here, like glucose and galactose together, we call it as globosides. Okay. And to that globoside, if then there is an additional N-acetyl neuraminic acid, which is also called as sialic acid, if it is attached, we call that as GM3 gangliosides. Gangliosides always contain N-acetyl neuraminic acid, which is also called as sialic acid. So we have GM3 ganglioside like this. If an additional N-acetyl galactosamine is added to this above structure, we call it a GM2 ganglioside. And an additional carbohydrate is added to this GM2 ganglioside, we call it a GM1 ganglioside. So ganglioside mono, GM for G for ganglioside, M for mono, and 1 for the position of this molecule in electrophoresis process. Heavier the molecules, it doesn't move faster. So fastest moving ganglioside is GM3 ganglioside. It will go to the third position, and then compared to the GM1, which is heavier, it doesn't move much faster. Okay. So this is how sphingolipids are. This is basically the overview of sphingolipid structure. Let me now take into the topic for today is degradation of sphingolipids. Okay, so the sphingolipid that is shown here contains two neuraminic acid molecules, one and another nana, and then we have galactose N-acetyl galactosamine, galactose, glucose, and ceramide. So basically, ceramide is attached with different types of monosaccharides and modified sugars and also neuraminic acid. So how this type of molecules are degraded? Let's look into that. All right. Very first enzyme. So basically the degradation of sphingolipids happens in the lysosomes of all our cells, lysosomes, and the most predominant organs where degradation of sphingolipids happens, it is in reticuloendothelial system present in the liver, spleen, and bone marrow. Remember that. And especially in those reticuloendothelial system, histiocytes and macrophages, basically the phagocytes, that's where lysosomes present in the phagocytes, that's where most of the degradation of sphingolipids will be going on. So basically the degradation of sphingolipids is a lysosomal degradation, and also lysosomal acid hydrolases, specific lysosomal acid hydrolases, they act sequentially on each of our sphingolipids and degrade, take one at a time, one molecule at a time in degradation of sphingolipids, which is happening in the lysosome done by lysosomal acid hydrolases, and we are going to talk about that now. So the very first enzyme here in the degradation is to remove this N-acetyl neuraminic acid. That's what is shown here. So neuraminidase enzyme is going to convert that into GM1 ganglioside. Now the GM1 ganglioside in the lysosomes, after removal of one of the N-acetyl neuraminic acid, it is acted on by an enzyme called beta-galactosidase, and this beta-galactosidase is going to break this beta bond between GalNAc and galactose, N-acetyl galactosamine and galactose, and remove this galactose. This galactose is removed, and that means GM1 ganglioside is now converted into GM2 ganglioside. Now the GM2 ganglioside has this N-acetyl galactosamine, which is attached with galactose by beta bond, and that is removed by beta hexosaminidase A enzyme. So basically beta hexosaminidase A enzyme is going to remove N-acetyl galactosamine, GalNAc, and note that if there is any deficiency of beta hexosaminidase A enzyme because of the mutation, it will give rise to Tay Schs disease. So in Tay Schs disease, beta hexosaminidase A enzyme is deficient and it fails to remove GalNAc, N-acetyl galactosamine, from GM2 ganglioside. That's why in Tay Schs disease, there will be accumulation of GM2 ganglioside. Okay, moving on. So the remaining molecule that we have here attached with the NANA, we have galactose, glucose attached with the ceramide. Now the NANA is removed by neuraminidase enzyme. So now we got ceramide attached with glucose and galactose. Glucose and galactose is a disaccharide. Basically, it's a lactose. So this is a cerebroside. Ceramide with disaccharide is a cerebroside. Now this molecule, it is basically the galactose attached with the beta bond is removed by beta galactosidase enzyme. Deficiency of beta galactosidase will give rise to Fabry disease, and that means there will be accumulation of cerebroside in Fabry disease, that is lactosyl cerebroside. So now once that happens, then we have ceramide attached with the glucose, so with the beta bond, and beta glucosidase enzyme is going to remove that. So there will be, if there is a deficiency of beta glucosidase, that leads to Gauchers disease. Okay, now we have this, for the previous one, Fabry disease is not because of beta galactosidase enzyme, it is because of deficiency of alpha galactosidase enzyme. So because we have this ceramide attached with the trisaccharide, glucose, galactose, it is a ceramide galactosyl ceramide or ceramide trihexoside, globosyl ceramide or ceramide trihexoside. That's the name for this molecule, ceramide trihexoside. Trihexoside means we have glucose, galactose, and galactose. Trihexoside, 3 hexosides attached to the ceramide. So this ceramide trihexoside is converted into cerebroside having 2 monosaccharides, that is lactosyl ceramide. So that will be done by alpha galactosidase enzyme, which is going to break alpha bond and remove this galactose, and you get cerebroside with glucose and galactose. That is done by alpha galactosidase A enzyme, and deficiency of this enzyme will give rise to Fabry disease. So in Fabry disease, what happens? So there will be accumulation of ceramide trihexoside, which is ceramide with glucose, galactose. Okay. Now coming to this side here, ceramide with glucose, beta glucosidase enzyme removing that glucose by breaking the beta bond. Deficiency of beta glucosidase giving rise to Gauchers disease. So if there is if beta glucosidase is working, so ceramide separated from glucose, so you get ceramide here. Now what happens to that ceramide? So the ceramide it breaks into sphingosine plus fatty acid done by enzyme ceramidase enzyme, which is not written here, ceramidase or ceraminidase. So deficiency of ceramidase or ceraminidase, it will give rise to Farber disease, not written here. So that's how ceramide separates into sphingosine and fatty acid. These are simple molecules and they can be reutilized by our cells. That's on this side where we have broken down GM1 ganglioside, GM2 ganglioside, and simplified that in the lysosome. What about like sphingomyelin? So the sphingomyelin, let me just remove this. Sphingomyelin, degradation of sphingomyelin. As I explained you, sphingomyelin is ceramides plus phosphocholine, that's a sphingomyelin. So we have a sphingomyelinase enzyme in the lysosome, which will separate that phosphocholine from ceramide, so releases ceramide here. Deficiency of sphingomyelinase enzyme in the lysosome, it will give rise to Neiwman Pick disease. Neiwman Pick disease. And Sphingolipidosis, I have a separate video of all these Sphingolipidosis. You can watch that video in my channel. Okay, that's how you get ceramide there. And what about if there is any attachment of sulfate in sphingolipids, which are sulfated? So the first thing is to remove sulfate, and that is done by aryl sulfatase enzyme. Aryl sulfatase enzymes remove sulfate and release ceramide with galactose. Usually sulfate is attached to the galactose. Deficiency of aryl sulfatase enzyme, it will give rise to metachromatic leukodystrophy. Remember that. Okay. Now, certain globoside you remember, it is a ceramide with different types of monosaccharides. So it's a globoside. Okay. Cerebrosides, globosides, especially if it has an N-acetyl galactosamine. See, there's an N-acetyl galactosamine here with the beta bond. Deficiency of beta hexosaminidase A enzyme and beta hexosaminidase B enzyme. Beta hexosaminidase A enzyme is going to take care of degradation of GM2 ganglioside, and also beta hexosaminidase B enzyme is going to take care of degradation of globoside. Now, if there is a deficiency of both hexosaminidase A enzyme and also hexosaminidase B enzyme, which is handling globoside, that will give rise to a disease called Sandoff disease. So in Sandoff disease, there is deficiency of 2 enzymes, beta hexosaminidase A and beta hexosaminidase B enzyme. That leads to accumulation of GM2 ganglioside and also globoside. Okay, so this is how degradation of sphingolipids happens in our body, especially in the lysosomes of our cells. So that's about degradation of sphingolipids. I hope this video has helped you in understanding a small concept here, how sphingolipids are degraded, and this will help you in understanding sphingolipidoses. All the sphingolipidoses I have mentioned in my video, and each sphingolipidosis video is available in my YouTube channel. You can watch it later, and also individual disorders. I will come up on that particular aspect. So if you haven't considered subscribing to my channel, so please subscribe to Dr. Mungli's channel for regular updates on the video. And also I have a playlist of each topics of biochemistry and even other medical related topics. If you're interested, please take a look at them and see if you need some of those videos, if they are interesting for you. So that's all from my side for in this video. I'll see you in my next video. Till then, you take care.